All right, let's get you ready for Professor Pasquinelli's final exam. Today, we're going to zero in on Lecture 14 and make sure you conquer every single concept so you can lock in those points.
So here's our game plan. We're going to break down the three big topics: microarrays, RNA-seq, and viruses. Then we'll wrap it all up with a super clear strategy to help you ace any questions that come your way on the final. Look, the whole idea here isn't just to rehash the lecture. It's to build your confidence by showing you not just what you need to know, but how Professor Pasquinelli might actually ask about it on the exam.
So for each topic, we're going to use a simple framework. What's the concept? What's a good example? And most importantly, how will you apply it? This way, we turn these ideas into actual points on your test. Let's get into it.
First up, microarrays. Now, this is a classic technique, and you can pretty much bet there's going to be a data interpretation question on this. So let's make sure you know exactly how to read those dots. All right, so let's quickly walk through the process. At its heart, you're just comparing RNA from two different situations. So you take your control cells, let's say cells grown without any serum, and you make cDNA that you label green. Then you take your experimental cells, the ones you treated with serum, and you make cDNA from them that you label red. You mix them both together, and you wash that mix over a special slide that has tiny bits of DNA from thousands of different genes stuck to it.
Now this slide, this is your cheat sheet for the final. Seriously, you absolutely have to know this color code. If a spot is green, it means that gene was way more active in your control cells. If it's red, it was more active in your treated cells. Yellow means it's pretty much expressed at the same level in both. And if a spot is black, well, that gene isn't really on in either condition.
Okay, so let's put this into practice with a question that feels like it's straight off the exam. You get an image of a microarray. You see a green spot for gene A, a red spot for gene B, and a yellow one for gene C. So what do you conclude?
Simple, right? You just break it down using the code. Gene A is green, so it's on fire without serum. Gene B is red, so adding serum clearly turns it on. It's a serum-responsive gene. And gene C is yellow, so it's probably not regulated by serum at all. Now here's a pro tip, something that will make your answer even better. Remember that the amount of RNA you see is a balance between how much is being made, transcription, and how long it sticks around, its stability.
Okay, so that's microarrays. Now let's talk about the modern successor, the current gold standard for this kind of analysis, which is RNA-seq.
A compare and contrast question between these two is a classic exam question, so let's nail the key difference. With microarrays, you're limited. You can only see the genes you already knew about and put on the chip. But with RNA-seq, because you're sequencing everything, you can discover brand new genes, find different splice variants. It's a much more powerful discovery tool.
And the process itself is really different. We're way beyond just seeing a colored dot here. You isolate all the RNA, turn it into cDNA, and then you sequence millions and millions of little fragments of it. Then the computer goes to work, taking all those little sequence reads and mapping them back to the genome. The more reads that match up with a certain gene, the higher its expression was. It's that simple. All right, third and final topic from Lecture 14, viruses. Now, for the exam, you really want to focus on two main things: their incredible genetic diversity, and maybe more importantly, how we've actually managed to turn them into powerful tools for medicine.
And this really gets to the heart of it. As Professor Pasquinelli said in lecture, the playbook for viral genomes is just wide open. They've tried pretty much every possible combination of nucleic acids.
And you've got to appreciate just how wild this diversity is. You've got your standard double-stranded DNA viruses, kind of like us. But then you have RNA viruses, like SARS-CoV-2, which has a positive sense single-stranded RNA genome. That means the second it gets in the cell, the ribosome can read it directly like an mRNA. And then you have retroviruses, like HIV, which do this amazing trick of turning their RNA genome back into DNA.
But, and this is so critical for the exam, underneath all that complexity, every single virus has to solve the exact same two problems. Number one, it has to make its proteins, and to do that, it needs a positive strand RNA that the host cell's ribosomes can read. And number two, it has to make more copies of its own genome to stuff into all the new virus particles. That's it. That's the whole game.
And that whole idea that viruses are basically just little machines for getting genetic info into cells leads us right to one of the coolest applications you need to know: gene therapy. We can actually hijack a virus, strip out all its nasty disease-causing genes, and replace them with a working copy of a human gene that a patient is missing. And the example from class for spinal muscular atrophy, or SMA, is a perfect illustration. These patients are missing a functional gene called SMN1. So scientists basically hired a harmless virus to act as a delivery truck. They load it up with the correct SMN1 gene, and it delivers its cargo right to the nerve cells that need it most. It's incredible stuff.
Okay, let's wrap this up and put a bow on it. Here's your final strategy, your game plan for any question from Lecture 14 that Professor Pasquinelli throws at you.
Here are the 4 things to drill into your brain. 1. Know those microarray colors cold. Red, green, yellow. What do they mean? 2. Be ready to explain why RNA-seq is more powerful than microarrays. 3. For any virus, remember its 2 jobs: make protein, copy genome. And 4. Connect that viral biology to the real-world application of gene therapy. If you've got those 4 points down, you are in fantastic shape.
And that's it. You've got this. We've walked through the key concepts from Lecture 14, and everything should be fresh in your mind. So the only question left is, which of these are you going to review first to really make it stick? Go crush that exam.